afternoon, friends. Welcome back. Am I audible?
Yes, ma'am. Good afternoon.
Okay, so fine. So this we are going to today we are going to start the fourth module, and which is about host pathogen interaction. Right. So in this we are going to study about microbial pathogenicity, one thing, and another thing, the defense mechanism in the plant, right? So when we talk about pathogenicity, so definitely it refers to all the factors which leads to the development of the disease, right? So it include the virulence factors of the pathogen, and which are different enzymes or toxins. They could be host specific, they could be non-host specific also, and certain growth regulators also, right? The first part we are going to discuss is about the virulence factors of the pathogen, and how the different virulence factors of the pathogens, maybe enzymes or toxins, they are being released by the pathogen, and they help in the development of the disease, right? And in the second part, I told that it is about the defense mechanism in the plant, right? So plants, they don't have a immune system like humans, like, or not, they don't have any developed kind of an immune system like animals or the humans, right? But yes, they there are certain concepts of constitutive defense mechanism in the plants, which include your inducible structural defenses and inducible biochemical defenses also, right? Structural defenses are like certain changes in the plant, or certain, for example, the corky layer in the plant, or abscission layers, or certain gum secretion in the plant, right? Which helps in the prevention of the pathogen. Or biochemical defenses or inducible biochemical defenses are hypersensitive response shown by the plant, or a systemic acquired SAR, systemic acquired resistance shown by the plant, right? So these are the basically defense mechanisms in the plant which are against the pathogen. So definitely they don't have any immune system or anything like that. But yes, they do respond to the pathogen attack, and in their own way, they try to do whatever they can do to prevent the pathogen attack, right? Or to just maybe just to stop the pathogen movement in the host or in the plant further. So they like a human immune system, yes, they do act in some way or the other way to prevent the pathogens, right? For example, the gene silencing. So whenever we gene silencing is the, or a post transcriptional gene silencing, which is called PTGS also, it is the best way of the plants fighting against the pathogens. They just try to shoo away the pathogen by using this mechanism, right? But then pathogens also, they need to evolve. So for example, the gene silencing mechanism in the plant is being shown against many viruses, it has been reported. But now we find that there are a number of the viruses who have evolved, and certain proteins, they act as a suppressors of the gene silencing, right? So ऐसा नहीं है कि सिर्फ plants evolve हो रहे हैं या उनका defense mechanism, but pathogens are also evolving, and they try to overcome that, the defense mechanism of the plant, in order to infect the host, right? So it is the both way, right? Plants are evolving, pathogens are evolving, so they are coming up with some other ways to infect the host, right? So gene silencing, maybe in a plant tissue culture, in one lecture we'll try to discuss how gene silencing can be used for the generation of some genetic transgenic plants also. The gene silencing mechanism can be used to develop plant or virus resistant plant. So there we'll discuss in gene silencing more detail. But in otherwise, gene silencing or a post transcriptional gene silencing is also a mechanism by which plants, they just try to kill the pathogens, or just try to stop their movement in the plant, right? Okay, so today's lecture we are going to discuss about what are the different enzymes which are being secreted by the pathogens, especially viruses and viroids. They don't secrete any enzymes, but enzymes or toxins, certain toxins are being secreted by the fungal or bacterial pathogens when they infect plants, right? So when we talk about pathogenesis, it means the development of the disease, or whatever events which leads to the development of a particular disease, right? It could be, changes in the structure or function of a cell or tissue or organ caused due to the microbial, chemical, or a physical agent, right? So whatever those events they together, this process is called pathogenesis. Again, this is a disease. The pathogenesis of a disease is a mechanism by which etiological factors they causes the disease. Term can also be used to describe the development of disease such as acute, chronic, or a recurrent. Usually, chronic, recurrent. These terms are used in a human diseases. And this word comes from a Greek word, pathos mean disease, and genesis mean creation. So there are several chemical weapons which are secreted by pathogens, which they utilize as they carry out their activities in the host. And these weapons could be enzymes or toxins or growth regulators or certain polysaccharides, right? So enzymes such as chitinases, cellulases, pectinases, and lignases are often secreted by the pathogenic organism. This is to gain entry into the plant, right? Fungi, nematode, and bacteria, they are all known to produce one or more of the above enzyme in the specific host pathogen or pathogen host combinations. But viruses and viroids are generally not considered to secrete enzymes. Although some viruses they may encapsulate enzyme in them. This is another process, but not as a defense mechanism here, right? So viruses and viroids they do not secrete any kind of the enzyme. So viruses and when we talk about the viruses, so usually PTGS is the mechanism which viruses uh, they reply to this PTGS mechanism by producing certain suppressor proteins which suppress the gene silencing. So this is how they overcome this, right? So this is a separate thing. So pathogenic organisms either continually they secrete enzymes or when they contact the host plant they secrete enzyme, right? So the cuticle, we know the cuticle is composed of there's a wax cutin which impregnates the cellulose wall, and the cell wall is also composed of a cellulose which again make up the structural framework of the wall along with certain hemicellulose, glycoprotein, pectin, and lignin. There's a number of compounds, right? So that means getting entrance into or penetration into the living parenchymatous tissue and then degradation of middle lamella is due to the action of one or more enzymes which degrade these chemical substances. So in order to get entry into the plant, they need to secrete certain enzyme which degrade the cell walls or the cell membrane so that they can get inside, right? So cutinases, they degrade the cutin on the cuticle layer, they pre-softening the tissues for mechanical penetration or as a first step in the tissue degradation. The cell wall is a structure which give a shape to the plant, which prevents any movement from inside or outside to vice versa, right? Nothing can go inside or outside that due to the cell wall. So definitely when pathogen, they need to enter a particular plant cell also, they need to cross that barrier, right? So definitely it need to do something so that it can get entry. So either maybe wounds due to some natural processes or environmental actions in the field, so they can get entry. Otherwise, then they need to secrete certain enzymes to soften up the cell walls, and they can get some entry into inside the cell, right? So studies have shown that several fungi and at least one bacterial species they produces cutinases enzyme, and there are a number of studies which shows that cutinases are continually produced in a low concentration with degradation products often inducing even higher level of cutinase secretion. And studies have shown that in some organisms, cutinase production may also be linked to the virulence of a particular pathogen, right? So how much it can cause a disease, how much virulent is the pathogen, it could be linked. Then there are cellulases. Again, we know the cellulose is one of the important molecule of a plant cell wall. It exists as a microfibrils with matrix molecules such as glycoprotein or a hemicellulose or a pectins or a lignins, right? Filling the spaces between microfibrils and cellulose chains. Again, cellulases or these enzymes, they are shown to be produced by pathogenic fungi, bacteria, and nematodes. These enzymes, they play a role in softening and disintegration of the cell walls. No doubt, these enzymes are involved in invasion and spread of the pathogen, but they are also instrumental in collapse of the cells in the tissue. So they are killing those tissues, right? They are softening those tissues. Indirectly, these enzymes participate in disease development by releasing soluble sugars that may be used as a nutrient by the pathogen. So this is important here. They are not only providing the way to the pathogen inside the cell, but the sugars that they are being released or soluble sugars, they can be used as nutrient by the pathogens and also may be involved in release of materials in the vascular system, interfering with transport or translocation of the water in the plant. So this is the another way it is affecting the plants. Then we have a hemicelluloses. Again, hemicelluloses are complex polysaccharide polymers which link the ends of pectic compounds to cellulose microfibers, right? So since hemicelluloses are, again, they are very diverse group of polymers such as xyloglucans or galactomannans or arabinoglucans, right? So several hemicelluloses have been identified in many plant pathogenic fungi. So the mechanism by which they participate in cell wall breakdown is not clear and is not known that how they contribute to pathogenesis, but these enzymes are being released. Again, then we have a ligenases, right? Lignin is a phenyl propanoid which is found in the middle lamella and secondary cell wall of the plant. So more than anything, it provides toughness or a woody nature to the woody tissues. It makes the wood hard. So again, around 500 species of fungi are capable of decomposing wood. Most lignin degradation is by basidiomycetes known as white rot fungi. So these are the fungi which are known to infect most these woody plants, and these fungi they produce lignases that enable the fungi to utilize the lignin. Then toxins, these were the enzymes, right? Enzymes, celluloses or hemicelluloses or lignases. Now toxins, right? Toxins are a chemical weapons of the pathogen. So toxins are again released by the pathogen. So toxins, they have been implicated in the plant disease as far as Anton D. Berry was the person who gave the toxin theory basically. Right, so the toxin theory is that a toxin is elaborated by a pathogen may produce all. So if we just use the toxin which is secreted by the pathogen, it can produce all the symptoms of the disease which is caused by the pathogen, right? So as more information was developed, the theory was largely, it was later discarded. They found that toxins may act directly on a living host cell. They can even damage or even kill the host. And some toxins are active on a wide range of plant species, non-host species also. And in some cases, such as toxin, which is Victorine, it is host specific. So they can categorize the toxins into a non-host specific also. And some toxic, they are host specific toxins also, right? So these are certain few toxins which are being produced by different pathogens. First is a TAB toxins. Again, this toxin is involved in a wildfire disease of tobacco, and in this disease the leaves they show necrotic spots which is surrounded by a yellow halo, right? The identical symptom of the disease may be induced by cultural filtrates of the organisms or by the purified toxins also, with symptoms which are identical to the wildfire of the tobacco. Now similar effects are also observed on large range of the hosts, so that means these toxins are non-host specific because they can produce the similar symptoms on the other non-host plants also. Then TAB toxins is chemically a dipeptide composed of the amino acids threonine and tabtoxinin. And in the cell, tabtoxin is cleaved, releasing the tabtoxinin moiety, which is an active toxin compound. The inhibition of the enzyme glutamine synthetase is the primary mode of the action of this particular toxin, right? So the TAP toxin is being released by certain fungi, and this toxin is a non-host specific toxic. That means it can affect the number of the host. It is not produced by a particular host only. Then we have a phaseolotoxin. Again, this is the toxin which is being involved in bacterial bean blights, which are called as a halo blights also. The symptom of the disease caused by the bacterium can be produced by the toxin alone. So if this toxin we are just inoculating on the plant, the similar symptoms are being produced. Chemically, the toxin is tripeptide or ornithine alanine. See, I'm just telling you the chemical structure to understand better, but you don't need to remember the chemical structures because it is difficult, I know. For the moment, you just remember the toxin, whether they are host specific or the non-host specific, and the diseases with which they are associated, right? Not, you don't need to remember the chemical structure here. So again, within the cells, the toxin is enzymatically cleaved, releasing phosphosulfinyl ornithine, which is the toxic moiety here. And cellular effects are a result of inactivation of the enzyme ornithine amylotransferase. So this is how it acts. Then we have a tentoxin. This is a very important toxin, and this is produced by Alternaria alternata. And this disease caused number of disease. This pathogen caused number of disease in the Alternaria leaf spots are a very common leaf spot fungi. Again, seedling death will result when greater than 1/3 of the leaf become chlorotic, and it will reduce the vigor with less than the amount of. So these are the symptoms. Basically, the toxin is a cyclic tripeptide which binds to and inactivates a chloroplast coupling factor. So that is why they become yellow and they become chlorotic, right? Then we have a host specific toxins also, right? So the idea that toxins play causal roles in plant disease is very, very attractive. And I mean, Anton Lever was the first person who started describing these things, right? So a very good reference on the role of toxins in the plant diseases, microbial toxin in a plant disease. It was basically published in an annual review of microbiology in 1963. And this review gives excellent account of discovery of a host specific toxins, right? So when one reduces the toxin theory to its most elementary form, it can be stated that all of the symptom of a given result from the direct action of the toxic product of the pathogen in that particular disease. So whatever symptoms are being caused are directly basically because of the toxins which are being produced by the pathogen. So they say that the toxins, when applied at concentration which could be reasonably expected around the disease plant, produces a susceptible host all the symptoms which are characteristic of a particular. This is how they tested it, right? The pathogen and the toxin exhibit similar or host specificity, so they are host specific. The ability of the pathogen to produce toxins varies directly with its ability to cause the disease. So that means if more toxins are produced, the more disease is being caused, and single toxin was involved in this case, right? So first toxin that isolated and fulfilled the above criteria was produced by Helminthosporium victoriae. So Helminthosporium leaves blight of oats. This is a disease which appeared in 1945 with introduction of number of. I discussed this last time भी मैंने शायद discuss किया था. Oat variety of Victoria के साथ ये disease हुई थी, which contained the VB gene for resistance to the crown rust disease. So in the Victoria line, the infects the basal portion of the plant. As it infects, the fungus elaborates the toxin which is carried to the leaves, causing severe leaf blight and the death of the plant. Then we have a T toxin. This is also a host-specific toxin. This is produced by southern corn leaf blight, which is caused by the fungus Coccoliobolus citrosporus, and this was earlier known as the Helminthosporium, right? The T toxin is produced by race T of the fungus, which is first appeared in United States in 1968, and it causes severe losses there. Corn varieties with normal cytoplasm were found to be resistant to the fungus and also to the toxin. The toxin, which is mixture of a linear long polyketones with 35 to 45 carbon atoms. This toxin affects mitochondria of the susceptible cell, where the ATP synthesis is inhibited by this particular toxin, right? So T toxin is another toxin. Then we have one HC toxin. This is again a host-specific toxin, which is being produced by the fungus Helminthosporium, which causes a leaf spot disease in the corn again. And this is only specifically to the certain maize varieties, not all plants. The mechanism is not very clearly understood, although the molecular and biochemical basis of the resistance is well understood for this particular toxin. So we discussed about the enzymes, then about the toxins. Toxins, host specific and the non-host specific toxins. Then growth regulators as the weapon. So we all know the growth regulators or a plant growth hormones, they are required in a certain amount to carry out the normal functioning of the plant, right? So the plant growth is a result of a cell division, elongation, and cell differentiation. And in some steps, somewhere the growth factors or growth regulators are important. So plant growth is controlled by naturally occurring compounds with hormonal actions that are referred to as a plant growth regulators or a plant growth hormone. So these are the common terms that we use: auxins, cytokinins, gibberellins, right? All these hormones. The major group of plant growth regulators are auxins, gibberellins, kinins, and the ethylene. So plant growth regulators, like a harm, we compare them. When in humans we use the term hormones, but when we talk about the plant growth or a plant, we use the plant growth regulators, though the plant hormones are also terms which are being used, right? So they act in a very small concentration and with slight changes in the normal level, resulting in a lot of changes in the growth pattern of the plant. So pathogens, which causes some pathogens, they causes imbalance in the hormonal system by causing infected plant to produce more or less hormones. So क्या होता है when pathogen attack or some in not in all pathogens but in some pathogens they create some imbalance in the hormonal system. So sometimes these infected plants they produce more hormones or sometimes they might produce the less hormones, or in some cases pathogen itself elaborates hormone thus changing the hormonal level, right? So definitely if they are affecting the hormones or plant growth regulators somewhere, so plant growth will be affected. For example, auxins are the major, we know they are the major growth regulators, and they play a role in a cell elongation, right? Indole 3 acetic acid. The naturally occurring auxin is a indole acetic acid or IAA. So increased level of auxin occur in a plant which are infected with fungi, bacteria, viruses, molecules, or nematodes. So this have been reported that there is a more auxin which is being released when these some pathogens they are infecting the particular plant, right? So they are, for example, corn smut, which is caused by Astilogomades, or club rot of crucifers, which is caused by Plasmidiophora, or southern bacterial wilt, or a Pseudomonas, or a crown gall, right? These are the good level of good examples of diseases which altered the auxin level in a which have been implicated in the disease. So these are the certain examples which changes the auxin level in the plants which are infected by them, right? Then gibberellins, right? So we know that how gibberellins were identified. So when we know a rice farmer in the Asia, they noticed some exceptionally tall seedlings which were growing in their paddy. And before these rice seedlings could mature and flower, they grew so tall that they toppled over, right? So they again, this aberration in a growth pattern was known as a foolish seedling disease of a rice in 1926. Right. Then Kurosawa, a Japanese scientist, discovered that this disease was caused by a fungal pathogen, which is a Gibberella fujikuri. So they discovered it late then, right? But then by 1930s, they found that the fungus produced hyperelongation of stem rise by secreting a chemical, which was the gibberellin, which was named as a gibberellin, right? So this is the gibberellin is the normal constituent of a green plant and is also produced by several other microorganisms. So best known gibberellin is a gibberellic acid, and there are around more than 80 different gibberellins which are occurring naturally in the plant, right? So spraying of a diseased plant with gibberellin overcomes some of the symptoms such as stunting, which is caused by several virus or molecular pathogens, indicating that gibberellin might be involved in the disease development, right? Then we have ethylene. So ethylene production again in infected tissue can be induced. This induction is again dependent upon the activation of ethylene biosynthetic pathways in the plant tissues, right? So genes which encode several key enzymes which are involved in ethylene biosynthesis are highly activated at the transcriptional level. So it has not been shown that ethylene is produced directly by plant pathogenic fungi or bacteria. So this is not being shown, but this show that ethylene production in infected tissues can be induced. Then ethylene has also been considered as a signal in plant for wounding and senescence response. So if plants are wounded, in that case also ethylene is being produced, or senescence in cases where plants are in the senescence stage, there also this has been produced, right? So recent studies shows that ethylene together with another signal component with jasmonic acid plays an important role in plant defense responses of several pathogen systems. So induced ethylene production in infected tissues. So infected tissues, ethylene production is being induced, right? So ethylene induces expression of certain type of PR, pathogenesis related genes, and some signal compounds which are involved in a defense signaling. Again, the ethylene sensitive mutant blocks several PR gene expressions, and ethylene insensitive mutant can be beneficial against some of the pathogens, right? But again, they are deleterious to the resistance against pathogen in a specific gene for the gene manner, right? जब हम plant में रेजिस्टेंस या डिजीज की बात करते हैं तो हम पीआर जीन्स की बात करते हैं, पैथोजन रेजिस्टेंस जीन्स और पीआर जीन एक्सप्रेशन की बात करते हैं, राइट? So there are a number of theories for a gene for a gene resistant, or for every plants have a certain type of a resistance genes set for certain pathogens. So there are certain theories which Vander Planck or some person they gave these theories, right? So there are certain amount of the resistance genes which are naturally present in a certain plants against some pathogens. So there is a concept of a gene for a gene. These kind of a concepts are present when we talk about the pathogen, but we are not going to study this in detail because it involves a lot of things which are not part of this syllabus, right? So basically what we discussed today in this lecture is basically that how pathogenesis is, or a pathogen, they produce certain enzymes to gain entry into the plant, how they growth regulators, how they produce certain growth regulators to gain entry into the plant, right? So these are the pathogen factors, that how when they affect certain hosts, they are producing certain toxins or certain enzymes, or how they are modifying the growth regulators for their own use when they are affecting the plant, right? Because see, not all pathogens cannot affect host plants, right? They can only affect if they are getting entry into the plant, or those host plant or those plants are susceptible to the pathogen, right? But if they are susceptible also, then also they'll need to at least get entry, or they need to get inside the plant in order to infect a particular plant. So lot of, as I told you in the beginning, that lot of processes are involved, lot of things are involved. They release certain enzymes, they release certain toxins, and then the third part is the growth regulators. They can modify the growth regulators for their own use, right? So this is the pathogen part, that how pathogen is using different techniques, or by which different ways, how it is using different ways to get entry and to get into the cells of the plant so that it can infect a particular plant, right? The second part of this presentation, which we'll discuss next time, discuss about the host factors, defense mechanism in the plants. What are the defense mechanism in the plants by which they try to see pathogen is using the mechanism, or different ways so that it can get entry into the plant, then counter to that, right? In response to that, there will be the defense mechanisms in the plants, or plants is going to use whatever things they have in order to stop the pathogen entry, right? So it could be some, for example, I discussed in the beginning, they could release certain gums, they can form certain corky layer around the plants, or there's a certain abscission layers or a tyloses are being formed so that they don't gain entry, or they could also release certain chemicals or inducible biochemical responses. It's hypersensitive response. So what is hypersensitive response? When any pathogen, it tries, it infects the plant, it has gained entry into the plant, and now plant know that pathogen is inside it, right? So what it is going to do, it is going to produce certain chemicals, or it is going to kill that area where the pathogen has tried to enter. That is a hypersensitive response. So that area will become dead or necrotic, so that the movement of the pathogen to the other part of the plant is stopped. This is a hypersensitive response, that it is trying to avoid the pathogen entry into the plant, right? So there's a number of ways by which plants they defend themselves, and then there are a number of ways by which pathogens, because pathogen, what pathogen will try? Pathogen will try to, or try to do whatever it can take to get entry into the plant and to infect the plant, to multiply there, and then after multiplication, or it will exit and it will try to infect some other host plants, right? So their unka main purpose hai to invade as much as host they can multiply as much. But on the other hand, the plant is trying to stop it, right? Plant will do whatever it takes to stop this pathogen. So it is like a the way our immune system acts against the pathogen. So similarly, plants, they have their own defense mechanisms which act against the pathogen or which try to stop the pathogen so that it doesn't affect them, right? So these are the defense mechanism of the plant. So we will not go further this today. So we'll stop here. If there's any question or anything which is coming to your mind, please ask your questions. लेकिन ये आपको पढ़ना पड़ेगा, बिना पढ़े आप नहीं याद कर पाओगे. Without going through it again, you will not remember this, right? So you need to remember this. You need to remember the types of the toxins. You need to remember what types of the enzymes are being produced by the pathogens with examples at least, right? So examples, enzymes, what are the types of the toxins, and which pathogens they produce these kind of a toxins. So you need to remember that also, right? I'm not saying that आप सब चीजें याद करो. For example, I'm not telling you to remember the chemical compositions or chemically how these toxins they are composed of. No, don't. But at least name and how they are infected, right? So that you need to know. Just a moment.
Ma'am, will this be coming in the test tomorrow?
No. टेस्ट का आपका आज का नहीं, लास्ट वीक तक जो मैंने कराया है ना वही आएगा आपको. So that means in the exam you will be coming till the module 3rd. This is the module 4, so I'll not give module 4. Module 3rd तक plant disease epidemiology तक आएगा आपका एग्जाम में, ठीक है?
Okay, thank you,
Fine.
ma'am.
So just don't leave it for now. Just leave it, right? So okay, we maybe 1 or 2 more lecture for this module, and then we'll come for the management. Management is also journal lecture. बहुत सारा portion आपने पहले किया होगा, but still we'll go through them one by one. And then specific plant diseases तो आपका वैसे भी it's in your presentation, right? So don't worry. With Feb and we are going to complete this syllabus. But again, I'm telling you, since this is a new course, you need to study. You need to go through the things because you might not have studied these things earlier. These are new things for you. Plant pathology is totally a new subject. You've studied about viruses, but not about fungus or not about bacteria pathogens, right? So definitely you need to again and again go through these things in order to understand them, right? एक बार पढ़ के नहीं समझ में आएगा, आपको पढ़ना पड़ेगा. You study. There are a lot of papers which are available on the internet about these kind of things. These are actually, I have this Agrios, GN Agrios is one plant pathology by GN Agrios. This is one book which comprises everything about plant pathology. But I have its hard copy. I don't have its soft copy. I'll try if I can get it. So this GN Agrios has everything so beautifully explained about plant pathology. So it is like everything we want to know about plant pathology is in GN Agrios book. So I have its hard copy, my personal copy in my office. So last time also when students they study, I gave them my hard copy in order to go through the topics that we discussed. So, but right now since you guys are taking online classes, so I'll try to look for its soft copy available from somewhere. So it is a very good book in a very simple language. They have tried to explain everything, history till management, everything. I might have some older editions. अब तो उसके latest edition आ गए होंगे, but a very good book to refer when we talk about plant pathology, right? Okay, students. So just a moment. Fine. So you've started working on that field work or not yet? एक दिन में नहीं मिलेंगे आपको सिम्टम्स. आप सोचो अगर एक दिन में सारी फोटो खींच के अपलोड कर दोगे, वो पॉसिबल नहीं है. हैव यू डन? किसी ने कोशिश की इसको शुरू करने की वीक की? नहीं की है? नो. 28 फरवरी है ना, बहुत देर से है. तो 25 फरवरी को स्टार्ट करोगे एंड 28 तक तो 15 प्लांट्स मिल ही जाएंगे, राइट? दिस इज व्हाट यू थिंक? यस मानसवनी.
Ma'am, I'll be starting soon. I think I got something like COVID, so I'm still recovering from that.
Oh, okay, you're not well still.
No, I actually, ma'am, I don't know what happened because my mother and sister had tested positive, and then I had all the symptoms, but
but not
after I
positive.
tested twice, both times it came negative. So I don't know
Oh,
what the infection was, but I'm just recovering from that. So
okay.
I think I'll start by this week.
Okay, no problem. I'm just asking everyone. I was trying to ask others also that see, if you just go out for a walk or maybe in a park or in just like that, just keep this thing in mind that you all have mobile phones these days. We don't need cameras, right? In our time when I did my MSc, I had to get they were especially the camera persons there when I did my MSc. So we used to do thesis work. तो फोटोग्राफ के लिए मुझे स्पेशली कैमरा पर्सन को बुलाना पड़ता था, फिर फोटोग्राफ लेनी पड़ती थी, और अगर वो अवेलेबल ना हो जब सिम्टम्स हैं तो इट्स गॉन गॉन फॉर द सीजन. So was the problem. These days we all have a mobile phone, and wherever we can see, we can click photos. So this is the easiest thing. At that time, मुझे I used to work on this fungus Septoria obesa on a chrysanthemum. That was my thesis title that time. So when I isolated this fungus, and when I saw it under the microscope, so I needed that photograph under the microscope. So they were not, no facility was available. So I need to prepare the slide, and I need to go to the city, and there was only one person that time who used to click the photographs under the microscope, and I used to get those photos. और वो भी आपका जब वो फोटो आता था ना, तब पता चलता था वो ठीक है कि नहीं, बिकॉज राइट नाउ तो आपके पास है ना सॉफ्ट, जो अच्छी नहीं है उसे डिलीट कर दिया, जो अच्छी है रख लिया. नो, वी नेवर हैड दिस ऑप्शन. So I literally, when they used to come in a print form, then we need to select, okay, now this is fine or not. Or so we used to click number of photos at that time. So since you guys have a privilege, right? So you take, whenever you go out, just keep this thing in a mind कि कुछ दिखता है, तो just click that photo, try to analyze कि क्या है. And it could be the weeds also. That's what I've told you, not the plants or vegetables or ornamental weeds, because viruses in very clear images. अभी मैं अपने society में आसपास देख रही हूँ. I can see very clearly this one weed plant, and typical yellowing symptom on that wheat plant. It is wild sunflower. साइंटिफिक नाम नहीं याद आ रहा है मुझे. इट इज वाइल्ड सनफ्लावर एंड टिपिकल येलोइंग. बहुत ही अच्छे से सिम्टम्स उसमें नजर आते हैं आजकल भी. सो आई एम श्योर ये वीड दिल्ली एनसीआर में सब जगह मिलेगा, एंड यू कैन इजीली गेट. एंड एजरेटम बहुत कॉमन वीट प्लांट है जो सब जगह मिलता है, और उस पे वायरस के बहुत अच्छे सिम्टम्स होते हैं. तो कुछ चीजें तो हैं जो बहुत कॉमनली और बहुत इजीली आपको मिल जाएंगी, राइट. फंगल एंड बैक्टीरियल में थोड़ा सा ढूंढना पड़ेगा, बट अपने घर पे जो वेजिटेबल्स या फ्रूट्स आते हैं ना, उसी को ध्यान से देखना. कई बार आपको पोटैटोस को देखना, पोटैटोस आते हैं आपको नॉर्मल नेक्रोटिक और उस तरह के सिम्टम्स उस पे भी नजर आ जाएंगे. आपके यहाँ घर पे वो आते हैं दिस लाइन्स, नींबू, राइट. उसके कई बार स्किन पे आपको कॉकी सिम्टम्स मिल जाएंगे. दैट इज ड्यू टू द सिट्रस, व्हाट क्रिसैंथेमोमस, राइट. तो सिट्रस कैंकर. सो बहुत कॉमनली बहुत सारी चीजें आपको टोमेटो स्पॉट टॉस्पो के सिम्टम फ्रूट्स दिस टोमेटो फ्रूट पे नजर आ जाते हैं. तो बहुत सारी चीजें आपको घर पे भी मिल जाएंगी. ऐसा नहीं है कि यू नीड टू लुक आउटसाइड. जस्ट ट्राई टू सी. यस देवांशी.
Ma'am, on tulsi leaves, it's kind easy to like see the black spots. It's common very
वो
in
कॉमन
basil
तुलसी पे,
leaves.
पता तुलसी पे वैसे पैथोजन ज्यादा रिपोर्टेड है नहीं है, बट यस कॉमन ब्लैक स्पॉट्स और वो सब ड्यू टू द इन्वायरमेंटल उससे भी होता है. और तुलसी वगैरह पे ज्यादातर कई बार इंसेक्ट इन्फेस्टेशन भी होते हैं. आपको जो भी लगता है आप क्लिक करो एंड put it there and explain the possible, what could be the possible reason. आप करो explain, no problem. This is your field work, right? So it could be right and sometimes it could be wrong also, but at least you tried. So this is open for everything, medicinal plant and everything. घर पे अगर आपके टोमेटोज लगे हैं, टोमेटो पे तो आपको वायरल के तो बहुत अच्छे सिम्टम्स नजर आ जाते हैं. टोमेटो प्लांट अगर घर पे लगे हुए हैं किसी के, जैसे किचन गार्डन में लगा होगा, तो वेरी इजीली यू कैन सी दीज काइंड ऑफ सिम्टम्स. स्पेशली दोस्त जो हाउसेज में रहते हैं, या फिर फ्लैट वालों को तो नहीं ऑप्शन है, बट स्पेशली दोज पीपल हु लिव इन द हाउसेज एंड दे हैव सम स्मॉल किचन गार्डन्स एंड सम. आप ऑर्नामेंटल में रोज में देखोगे, अगर आपका रोज ग्रो है उसमें, दिस इज रोज मोजाइक वायरस और एप्पल मोजाइक का जो दूसरा स्ट्रेन है, रोज मोजाइक वायरस के बहुत कॉमन सिम्टम्स उसके लीव्स पे नजर आते हैं आपको. तो आई वेरी इजली, आपके पपाया लगा होगा किसी के घर के आसपास, पपाया लीफ कल एंड पपाया रिंग स्पॉट तो दिल्ली एनसीआर में हर पपाया ट्री पे है. हर in my society I these symptoms very clearly. So you just need to see. सब मिलेगा, बस you just need to see. थोड़ा सा interest के साथ you will get it, right? Okay, fine. So we'll stop here. Tomorrow you have exam. Prepare for exam, and I'll take attendance now. Switch on your cameras for attendance, students. Adhish Mishra. Adhish, are you there? Ayushi Jain.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi.
Present, ma'am.
Ishita.
Present, ma'am.
Janvi. Yeah,
Present, ma'am.
Manaswani. Namrata.
Present, ma'am.
Namrata is there. Nandini.
Present, ma'am.
Nehal. Nehal Joshi. Rupal Joshi.
Present,
Nehal
ma'am.
is right somewhere. Sahiba.
Present, ma'am.
Sanjana Sharma.
Present, ma'am.
Shakti.
Present, ma'am.
Shivani Mishra.
Present, ma'am.
Tanvi Sharma. Tanvi is there. Okay, Tanvi is there. Tridev.
Present, ma'am.
Tridev is there. Vivek. Yukta. Vivek and Yukta. Nehal is present. Adhish is present. Okay, Yukta, I can see you. Vivek, I did not hear you. Vivek, are you there? And
Ma'am,
Ikshit
he
is-
is having network issue.
हाँ, और देवांशी को पता चल गया और मुझे पता ही नहीं चला. See?
Ma'am, actually, in last class also, he was
Yes, ma'am.
facing issue. There was a power outbreak.
Okay, and Ikshitiज क्या क्लास में था क्या? Ikshitiज नहीं दिख रहा है मुझे. Okay, fine. Anyways, okay, students. So we'll meet tomorrow. We'll have exam first, and then we'll have a presentation. Okay, fine. Okay, bye bye. Then I'll see you tomorrow.
Thank you, ma'am.
Thank you, ma'am.